Hi everyone, welcome to Genetics. I am Dr. Rob Rooker. I'm a professor at the University of Minnesota, and I teach, mostly I teach in Majors Biology, our Foundations of Biology course, and I also teach, of course, in genetics. And I'm the author of your textbook, and I've prepared lectures for all the chapters, and so your instructor may be occasionally or largely using my lectures to help you learn genetics. The first chapter is an introductory chapter, and there's two kind of goals in this chapter. One goal is just to provide you an overview of genetics, and then another goal is to talk a little bit about some of the features of the textbook and the aspect of kind of the science behind genetics. So we're going to talk about those various things, so let's get started. So what better way to start genetics than with an overview of the Human Genome Project? It started in 1990. The aim was to decode our genome, all the DNA found within our chromosomes. This project was coordinated by the NIH and the DOE, and it was carried out by scientists from all around the world. Largely in United States, Europe, Japan, but countries from all over the world actually. So what the outcome was is kind of schematically shown here. Our bodies contain trillions of cells. Most cells are what we call somatic cells, like liver cells and skin cells. And in the case of humans, each of those somatic cells has 46 chromosomes. They're found in 23 pairs, so you got 23 from your mother, 23 from your father. If you stretched all the DNA out, it would be 2 meters in length. There's approximately 22,000 protein coding genes. That number is still changing a little bit, but roughly in that ballpark. And there's also many genes that don't encode proteins, like genes that encode transfer RNA, for example. The total amount of DNA in one set of chromosomes is about 3 billion base pairs of DNA. And as we're going to talk about, especially when we get into the molecular genetics part of the textbook, the structure of DNA lends itself to being organized into genes, and the DNA is composed of 4 different bases, which are abbreviated A, T, G, and C. So the completed sequence was published in 2003. Like I said, nearly 3 billion nucleotides per set. The accuracy was greater than 99.99%. Today it would be even higher. More recently, researchers, as we've gotten better and better at sequencing DNA, what researchers have been interested in terms of the human genome is comparing different individuals. So the 1,000 Genomes Project was launched, and the goal is to determine the variation in human genomes. And there's not a whole lot, actually. We're very, very similar. All people across the planet are very, very similar to each other in terms of their genome percentage. And it helped us determine how many genes we have and so forth, and how cells develop into complex tissues. One of the big goals of the Human Genome Project and the 1,000 Genomes Project is to consider how mutations in genes and genetic variation contribute to human diseases. And in the United States, a lot of this was funded by the National Institutes of Health. So while trying to understand genes and their function, scientists have developed different genetic technologies, and some of them are controversial. One example is mammalian cloning. In 1997, Wilmot and colleagues cloned the first sheep named Dolly. And more recently, cows, mice, pigs, goats, and cats have been cloned. There's fears that the technology might be applied to humans, and so this has led to some legislative bans on human cloning. Genetic technologies also allow researchers to modify traits in animals in various ways. For example, mice can be made to glow green. And what's going on there is a gene from jellyfish encodes a green fluorescent protein. And if it's introduced into lab mice, they look green when their light is shown on them. So this is Dolly. Dolly's the cute sheep here in the front, and that's her surrogate mother. And this is that green glowing mouse. So this is a newborn mouse that doesn't have fur, and so you can the fur does not actually glow green. It's just their skin where the GFP protein is being made, the green fluorescent protein that comes from jellyfish. Okay, so the first section that the chapter discusses sort of the basis of the molecular expression of genes. So genetics, in a general sense, is the study of heredity and variation. And along with evolution, it's really the underlying discipline in biology, because genetics, our genes are the blueprint for the construction of cells and the construction of multicellular organisms. Genetics is largely centered on the study of genes. The classical definition of a gene is kind of vague. It's defined as a unit of heredity. The modern definition that's more molecular is that a gene is a segment of DNA that produces a functional product, such as a polypeptide. So what genes do is they provide the blueprint that determines the traits of an organism. So largely our traits are determined by the proteins that we make. And the proteins that we make, the information to make those proteins is found in our genes. So that's how it works. The genes are really an informational storage unit, largely to make proteins. Then those proteins do all kinds of different things, play structural roles and enzymatic roles and all kinds of roles to make our cells. And what we see are traits in living organisms, and the traits are largely rooted in the expression of lots of different proteins. So if we take this down to the biochemical level, all cells are constructed from small organic molecules. These are linked together by chemical bonds to form larger ones. So the four main types of molecules are nucleic acids, DNA and RNA, proteins, which are composed of polypeptides, carbohydrates, and lipids. So living cells are composed of biochemicals. Nucleic acids, proteins, and carbohydrates are termed macromolecules. So they're polymers that are constructed from smaller molecules called monomers. And then cellular structures form as a result of the interactions of molecules and macromolecules. So let's just go through an example here. Nucleotides form the building blocks that are used to make long strands of DNA, which associate with proteins to form chromosomes. Chromosomes are contained in the cell nucleus. So here we're down at the biochemical level. These are nucleotides. These are organic molecules. They're linked together to form DNA, which is a macromolecule. Proteins are also macromolecules. They associate with the DNA to make a structure that's called a chromosome. And then chromosomes and other stuff like membranes that are composed largely of lipids are organized to form a cell organelle. And then this organelle, many organelles and other structures come together to form a whole cell. So this would be a whole cell here. So it's composed of organelles, and if we go back down, an organelle has got lots of macromolecules and molecules. This is one structure that's composed of macromolecules, and then we're back down to the biochemical level. So this is sort of the hierarchy of how life is organized. As I mentioned before, proteins largely determine cell structure and function. So each cell contains many different proteins that determine its structure and function. As far as terminology goes, a polypeptide is composed of a linear sequence of amino acids. So very technically, genes encode polypeptides. A protein is a functional unit that does something, like it might be an enzyme. So a protein can be composed of just one polypeptide, or it can be composed of two or more polypeptides. And I keep repeating this, but it's an important thing, is the characteristic of a cell largely depends on the proteins it produces. All of the proteins that a cell makes at a given time is called its proteome. And proteins have very diverse biological functions. Now we're going to think about the genetic material. So the genetic material in all living organisms is DNA. It's true that some viruses use RNA, but viruses are not considered living. They're not made of cells. But some viruses use RNA as their genetic material. So DNA contains genes that encode the information required to synthesize all cellular proteins. So it's able to do this because of its molecular structure. DNA strand is a polymer of nucleotides. It's double-stranded, and each nucleotide contains one base that has nitrogen in it, so it's called a nitrogenous base. So it's the four bases that can be found in a nucleotide. So there's only one base in each nucleotide, but there's four different possible bases that could be in a nucleotide, and those are adenine, thymine, cytosine, and guanine. As far as the storage of information goes, it's stored in the linear sequence of these bases. So your 3 billion base pairs of DNA is parceled into segments called genes, and each gene has a unique sequence of bases. And the sequence of bases in protein coding genes determines the amino acid sequence in a polypeptide. So, for example, if the DNA sequence is that's shown here, the amino acid sequence would be this. So ATG in the DNA would be AUG in the messenger RNA, and that would encode methionine, which is abbreviated MET here. GGC in the DNA would correspond to GGC in the RNA, and that would encode glycine. CTT in the DNA would encode CUU in the mRNA, and that would be leucine, and then this would be serine. So the DNA is sitting there storing this information. It's accessed by being copied into RNA, and then the information in the RNA is used to make an amino acid sequence in a polypeptide. And so this is just another example. So this is a different DNA sequence, and it would encode a different amino acid sequence. And so each protein coding gene just has a unique set of bases that encode a unique amino acid sequence for the polypeptide it encodes. At a larger level, genes are found within structures that are called chromosomes. Humans have, the somatic cells of your body, like skin cells or muscle cells, have 46 chromosomes. Each chromosome is a complex of DNA and proteins. The average human chromosome has about 100 million nucleotides per strand, so it has, since it's double-stranded, it has 100 million base pairs. An average human chromosome has about 1,000 different genes. So roughly, there's about 22,000. It's a little less than 1,000 on average, but there's 23 pairs of chromosomes, and there's about 22,000 genes. So it's slightly less than 1,000 per chromosome. So this is showing the human chromosome, two sets of human chromosomes. So this would be a human male. So this chromosome number 1. So one copy this male got from their father and one from their mother. This is chromosome number 2, and so forth. So each pair of chromosomes is found in homologous pairs, and that's because one came from the mother and one came from the father. What gene expression is? There's different ways of thinking about it. You can think about it at the molecular level, and you can think about it at the level of traits. But basically what it means is the information within the DNA is accessed in a living cell, and that's going to change the property of the cell at the molecular level and may affect the traits of an individual. So gene expression occurs in 2 steps. The gene is transcribed, so the genetic information in the DNA is copied into a nucleotide sequence of RNA. So DNA sits there and stores the information, and RNA is kind of accessing that information. And then during translation, the sequence of bases in RNA provides the information to make amino acid sequence of a polypeptide, but then the polypeptide becomes a unit within a protein. And so this is a picture of it. This is a gene. It's found within DNA. So this would be this DNA segment would be much longer, and it would be found within a chromosome. The gene is transcribed, and that produces an RNA molecule, which, as we'll talk about in many places in later chapters, makes messenger RNA. And then this messenger RNA is translated into technically a polypeptide. It says protein here. If a protein was composed of one polypeptide, it would be this simple flow. A lot of times proteins are composed of two or more polypeptides. And as I keep reiterating, the functioning of proteins largely influences an organism's traits. Okay, this portion of Chapter 1 has been largely focused on kind of more at the molecular level. In the next section, we're going to also be thinking about gene expression, but it's going to be more at the level of how does the expression of a gene affect traits in multicellular organisms like animals and plants.